Hello, friends. Good evening, and it's beautiful to welcome you back to our channel. Today, we're going to do a longer video where we just talk about culture-based techniques. Now, it's important in microbiology that we're able to grow microorganisms in the laboratory. We're going to make this a series where we'll be talking about different methods of culturing microorganisms and how we can run it in the laboratory. But it's important to note that culture-based techniques refers to a group of methods in which in the laboratory we're able to grow microorganisms, bacteria, viruses, and some parasites. We're able to grow them under controlled conditions. So we simulate conditions that encourage the growth of these organisms. And to make this possible, we have what we refer to as culture media. So these are chemical preparations, that are used to improve and encourage the growth of different kinds of organisms. Usually in the laboratory, we do a lot of culturing of bacteria. Different bacteria are cultured from different samples. So the idea is you have a sample, you're interested, you're wondering what are the kind of microorganisms, bacteria that are growing in this sample. So you have your initial preparation, you prepare, process the sample, and prepare it. Different samples are processed in different ways, to be able to then culture microorganisms out of these samples. And these cultures are important because the reason why you want to do this is that you want to be able to study these bacteria, study these microorganisms under different conditions. You want to be able to determine what are their energy sources. Do they grow under oxygen? Do they not grow under oxygen? So there are various questions that you want to be able to interrogate these organisms. And for you to be able to do these investigations, you must have what we refer to as pure cultures of these microorganisms. And what are pure cultures? This is cultures that you have from a single population, cultures that are offshoots, they're offsprings, they're progeny of a single organism. So you have an organism being proliferated into many, many different organisms, and then you're able to interrogate all these organisms. This is what we call a pure culture. So when we create a pure culture in the laboratory, then we are able to interrogate this pure culture. And many a times, a lot of work that we have to do is first of all trying to identify this organism, and we have different methods that we use to try and identify this organism in the laboratory. Now we know that we have controlled conditions under which we're able to grow these microorganisms in the laboratory. We want to control the temperature, we want to control pressure, we want to control the nutrient source, and then we're able to grow these microorganisms. And these are very important. And like I said, we have different samples. You can have environmental samples, you can have samples from soil, from water, from fecal matter, from food, from plants, from animals, from flesh. Different sorts of places where you can get samples from. You even do samples where you do swab samples. And what swab samples? You have a piece of cotton wool that you've soaked in either distilled water or normal saline, and you use it to gently rub on a surface because you want to take the microorganisms from that surface and go and culture them in the laboratory. So you can have samples from different kinds of places. And remember that microbiology is in different aspects. There are different aspects are Greek microbiology, medical, aquatic. So we keep taking samples from different places and then culturing them in the laboratory. I'll do a series of short videos this week where we'll talk about different methods of culturing microorganisms. Remember to like, share our content, and do have a blessed week. Thank you for listening.